Hello, I'm Yoon Dom Suk, a research assistant professor in the orthopedic class. Today, I am going to talk about the role of E3 ubiquitin ligase in osteoclast differentiation and its potential as a target for gene therapy. His will be about how E3 ubiquitin ligase, called pelino-1, which we studied, can influence on the undifferentiation process of osteocluster. Also, this will cover our study on whether it could play a role as a gene medicine for bone disease in the future. Firstly, osteoporosis is a bone disease occurring when a person's bone density is decreased. As getting older, most adult-age people have their bone density decreased. This symptom is notable among women in post-menopause. If you see the market outlook shown here, from 2014 to 2019, we do not see any big prospect within these years. From 2019 to 2024, it is estimated by the Health Industry Brief that the scale of global market in therapeutic for osteoporosis field will rapidly grow up to $9.3 billion. I think this is probably a phenomenon as the aging population increases, and the purpose of the current study is to develop drugs that meet the current trend. As I just mentioned, one of the risk factors for osteoporosis can be age. But as we age and various technologies evolve, we tend to have an inactive lifestyle, and this can cause osteoporosis. And being lack of some kind of thing can also be a cause. There are reports that approximately 1 in 3 adult women and 1 in 5 adult men in their 50s will experience fracture associated with osteoporosis. You could prevent osteoporosis, in some sense, by taking sufficient rest, eating things like calcium or vitamin D, quitting smoking or drinking. But when you already have an osteoporosis, you can only treat or prevent it with various medicines. We first need to learn about some of the biological factors that can develop osteoporosis before conducting an osteoporosis treatment development study. As shown in the illustration, osteoclast plays a role of rejecting bones, and osteoblast plays a role of rebuilding bone parts that are broken. These two cells balance bone homeostasis as they destroy and rebuild bones, which can go to osteoporosis if the balance breaks. As you can see in the picture on the right, osteoclasts' number may increase with age. But it's known that as mesenchymal stem cell ages, which can differentiate into osteoblast. It differentiates more to adipocyte, which is a fat cell, than osteoblast, and it breaks the balance and can become a cause of osteoporosis. Most of the drugs currently being developed inhibit these osteopath activities or increase osteoblast activity. However, in the case of bisphosphonates, which are currently the most popular, it has been reported that administration with long-term can interfere with bone formation, and some side effects in the jawbone and other areas have been reported until recently. In addition, many reports have been reported about newly developed drugs about its use, high cost, and side effects. For this reason, new drug development is currently being required in the growing osteoporosis market. We aim to develop a gene treatment called gene therapy among various treatment methods in the lab. To explain a simple concept of gene therapy that I'm interested in can treat genes by transferring DNA gene that's coding in a transferable vector to target tissue, or transducting or transfecting these to target cells from ex vivo and putting these back in the body after culturing. As you can see in the table, various target genes have been studied in osteoporosis field, and various vectors or cells for transferring genes have been used as candidates for treating osteoporosis. We first selected pelino-1 as a target gene and a candidate for gene treatment for osteoporosis. To explain about pelino-1 briefly, it is well known as E3 ubiquitin proteins ligases. The main role is known to regulate the immune response well by interacting with various vectors involved in the innate immune signaling pathways. In the case of this gene, the gene is not large, so when the gene is inserted into any particular vector, it is easy to insert, and that's why we selected this gene for our experiment. There are many related patents filed or registered in Korea and abroad if you search pelino. However, most of them have been tested as control factors related to cancer or inflammation, and those are the ones that are patented. We are doing research related to osteoclast differentiation or osteoporosis, and since there has been no reports of these, it is a new thing to develop a treatment for osteoporosis with Pelino-1. Let me tell you the results of the experiment. What we initially noticed is that osteoclast activity is known to increase with age. And this is the result of experiments that we wanted to observe the phenomenon, thinking the increase in osteoclast activity might be related with protein called pelino. As you can see in the A and B pictures, when we extracted cells that can differentiate to osteoclast called BMM from old mice 4 weeks ago and 10 weeks ago and differentiated to osteoclast, we were able to see that they were actually much more efficient in differentiation to osteoclast in the cell extracted from old mouse, and related activity increased as well. Pelino has a total of 3 subtypes. The reason why we were interested in Pelino-1 was because it not only showed a rapid difference in expression, but Pelino-1 compared to Pelino-2, 3, especially during osteoclastogenesis, compared to relative expression in young mice and older mice, by confirming that it is statistically significantly reduced in older mice. We thought that the reduced Pelino-1 levels, as we aged, were not associated with an increase in osteoclast's activity, and conducted the following study. We've also checked a result in protein. We've seen that protein level of Pelino-1 decreases rapidly as related osteoclast marker increases when it was differentiated to osteoclast. That's why we created a mouse to learn about Pelino-1's role in in vivo. As you can see in the picture on the left, we first created a Pelino-1 plus mouse. Got K-Cree mouse for the gene emerges in osteoclasts specifically. It's called Cathsin K. And we joined these two and produced a mouse that could only knock out only in osteoclasts specifically. Then, over the 6 weeks of birth, micro-CT was taken to observe bone density between the two groups. And as you can see in the picture C, we were able to see that the size of a mouse were small when the Pelino-1 knocked out osteoclast, specifically because of this bone density. As you can see in the picture B, the weight was significantly different between the two groups statistically. So, we thought Pelino-1 would be related to the increase of activity in osteoclast, even in vivo, because affects bone density when it was knocked out by osteoclasts specifically. We extracted monocyte from 2 mice. One was not knocked out, and the other one was from a mouse with Pelino-1 knocked out. As a result, in the picture A, we were able to see that the monocytes had greater ability to differentiate, and related trap activity could increase much more when it was knocked out comparing to groups. In the picture B, we performed trap staining to see the difference between osteoblast and osteoclast that's under the growth plate in femur. And we've analyzed this part mainly as osteoblast and osteoclast under the growth plate are most active in the part. As you can see in the picture, we were able to confirm that Pelino-1 had a significant increase in osteoclasts number in the femur of knockouted mice, and quantitatively it was also meaningful. Next, we also experimented with the level of CTS, known as any experimental marker of osteoporosis, and compared it between the two groups through ELISA, and we found that the level of CTX1 in the experiments in the Pelino-1 knockout mouse was much increased, so that Pelino-1 played a big role in increasing the osteoclast specialty knockability. Next, we wanted to see what the normal mechanism is and how Pelino-1 can regulate the osteoclast genesis of the mouse monocyte. In fact, this data was designed to analyze detailed mechanisms through RNA expressions, and not long after the RNA expression ended, we needed to do additional analysis, and once we had only a few representative results. We are primarily interested in analyzing a protein called NF-ADC1, which is one of the transcription factors that plays an important role in osteoclast eruptions. If you look at the number of illustrations, you could see that when you overexpression Pelino-1 or knock down through sernal, you could see that some transcription activity in NF-ADC1 was falling. or going up, and as you can see from the Western blot results in B, the protein levels of NF80C1 were actually increasing very much in the Pelino-1 knockout mouse. When we first identified that it was associated with osteoclast eruption through KEGG pathway analysis through the RNA expression in C, as you can see in the red box, we were able to see that there was a statistically significant difference in pathways involved in osteoclast differentiation between the actual two groups. And when we checked some of the key target MRI expressions, we were able to say an increase in the RNA of genes that work meaningfully in the monocytes drawn from the conditional knockout mouse. However, in the case of NF80C1, there was no difference in MRI levels because the Pelino-1 we are studying is a source translation regulator. So if you can adjust it directly, you can say that you have achieved some corresponding results because the MRI level is not controlled, but only the protein level, and I think we will further study how Pelino-1 can count on osteoclast genesis through further analysis and more detailed mechanism studies. And there's something we're further working on with the preliminary study, which is currently underway to determine what role Pelino-1 may play in bone differentiation on osteopenic differentiation on the mesenchymal stem cell. The reason for this is that MSC in the bone marrow, as seen on the left, will count and engage a significant part in the reconstruction of the bone. But as we age, the MSC is more biased towards adipocyte, and at the same time, the osteoblast differentiation rate decreases as the MSC ages. The activity itself that can differentiate into osteoblast decreases. These symptoms can also be a biological factor in osteoporosis, independent of an increase in osteoclast activity. As you can see, when you knock down Pelino-1 using the lentivirus of Pelino-1, you could see through a calcium staining called alizanin red S staining that bone differentiation is reduced in MSC, and it came out statistically meaningfully when quantified in several parts. So what we're seeing at the conclusion of this study is that while the experimental results are not yet perfect, Pelino-1 is concluding that osteoblast differentiation can be promoted, osteoclast differentiation can be suppressed, and if these things prove experimentally well, may be targeted as one gene therapy that can have dual effects or dual functions in treating certain osteoporosis. When we think of it as methodal, there were a lot of questions about how to pass Pelino-1 to target tissue when we tried to apply Pelino-1 in practice. Although the current mechanism study is ongoing, that's why we're interested in viral vectors, among them an AV virus called adeno-associated virus. Ultimately, we are experimenting with the purpose of inserting a gene named Pelino-1 into a specific AV vector that can target bone and passing it to target tissue. I recently signed an MOU with Professor Guangping Gao of the Hooray Gene Therapy Center at UMass, known as the Master of Gene Therapy. That's why we've developed the AAV9 vector, which can only target bone specifically, and vector has a peptide that can bind to bone. This vector has been introduced to a variety of papers through many experiments, and we plan to source it from the MOU-bound lab. We're going to put the genes we're doing in Pelino-1 or other labs of interest to see if we can actually target them with bone, see the therapeutic effects, and reduce the side effects because we don't go to other tissues. As it is a gene therapy towards the application, I think there will be a lot of challenges in getting to osteoporosis treatment right away. So what we're thinking about at the beginning is that once the gene therapy that Pelino-1 can be overrepresented in is completed, we can experiment with rare disease experimental models, and then try these things by applying them locally to osteoporosis fracturing patients if the results are good in the future. Then, if vector is further developed and secured, I'm thinking about whether it could be a target as a gene therapy that can be applied to people with systemic osteoporosis. Thank you for listening.